Can you hear me okay? All right. Hi, thanks for coming to the talk I'm going to give about the big story of little life. And anybody tuning in from their staterooms, hello. Thanks for listening, or if you're watching later on the recorded Viking TV. My name is Allison Cusick, and I was going to break this talk up into two parts. So first I wanted to tell you a little bit more about myself and my research and the types of science that are done on board the ship. And then the second part, I'm going to hopefully wow you with the wonders of the microscopic life that lives in the ocean and why I became so obsessed with it. Maybe you'll walk away also feeling an obsession, a newfound obsession. So, as I said, my name is Allison Cusick, and I am a researcher at Scripps Institution of Oceanography in San Diego, California, in the U.S., where it's sunny all the time. And in this last March, 2022, I was actually on board the Octantis with my colleague, Martina here. Martina is a researcher at Universidad Nacional de La Plata in Argentina. And she and I have been working together for almost 6 years now. It's one of those weird life moments where you realize one of your colleagues is going to be a life partner, and we have many plans to do research together for decades to come. So it was really fun to be able to get into the field and come on board the new ship, and then also to be back here on the Polaris. And then the difference between her and I, aside from the fact that I'm tall and she's short and I live in the US and she lives in Argentina, is that I am focused more on the genetics and trying to understand how microscopic life responds to a changing environment at the genetic level. And then she's a traditional taxonomist, which is somebody who can look under a microscope and just like that identify different species based on what they look like. So I think she has a superpower and she thinks I have a superpower. But we do a lot of work together, so I thought I should introduce her. And how did I personally get into polar science? I'm specializing as a polar oceanographer and a biologist. But it's biological oceanographer. Polar biological oceanographer is a mouthful. So I was hooked in Antarctic research when I first went down to the Ross Sea, which is, if you were to come straight from New Zealand, the Ross Sea is on the other side of Antarctica. And it was the very first time I had actually gone to sea. I was slated to work on one of the United States research vessels, the Nathaniel B. Palmer, for a 53-day expedition, where we lived on this ship, and this green stuff growing in the pancake ice is algae, or maybe if you saw Brandy's talk, have been hearing about diatoms, sea ice diatoms. And the goal of this 2-month expedition was to trace the fate of algal carbon as it exports into the seafloor in the Ross Sea. And then the growth in the surface, how does it change through the water column as it's going down to the depths below and feeding the animals that live on the seafloor? But also, how do the algae influence the clouds that are forming in the polar regions? So it was this full cycle that a team of scientists were trying to understand. And I actually have applied to be a NASA astronaut twice now and have not been selected yet. But when I landed in Antarctica for this first time, I was like, this satisfies that itch to go to another planet. It is literally extraterrestrial feeling. So I'm very content. If NASA called me now, I might say, no, sorry, NASA, I'm an Antarctic researcher. So we'll see. But definitely was hooked. And when you're living on a ship, maybe this ship, you might not get the sense of needing to keep morale high, but we all did experience it collectively during the beginning of the pandemic. A sense of fun and keeping yourself sane when you have nowhere to go, nowhere else to be. And so we bring costumes on board, and scientists have a lot of ways to stay humored. But that's where I decided I need to learn everything I could about this place. And if you haven't yet been to Antarctica, I always thought it would be this flat white expanse like you hear about with the South Pole expeditions. But when you're on the coast, this is video footage from the peninsula where the ship's going to land late October. This is showing the fjord system. So you have these mountainous peaks coming straight out of the water, glaciers that hit the ocean. They're called tidewater glaciers. I'll just play that again. And that's a really cool place to study how ice and ocean interact. A lot of these float on the ocean, so you can also look at how the ocean's affecting underneath these glaciers. And then, of course, they were all carved out from the past glaciation event, which is why you have these deep basins or fjords, and there's circulation patterns within the coast. And one of the really cool things that happens in Antarctica that is different from the glaciers, glaciers are the fresh water of snow accumulation, but in the winter the sun disappears and it causes the surface of the ocean to get so cold that it begins to freeze. So this is sea ice formation. In one of another talk earlier this week, Brandy showed that awesome circulation diagram of how all the oceans are connected to the Southern Ocean. So a lot of that formation of that deep water is here in Antarctica. With the formation of sea ice, you're rejecting all that salt, and it's very cold and heavy and dense, so it sinks to the seafloor. And I consider the Southern Ocean around Antarctica as the mother of all oceans. So it is all connected. One of the other things that happens after the winter with this sea ice formation, the sunlight returns and you start to get this growth of life. So this is the algae or microalgae. We also call it phytoplankton. And a particular type of phytoplankton that lives in the sea ice are called sea ice diatoms. So this is a bloom that starts to happen underneath that sea ice. And that is hard to see if you don't have a microscope, but under the microscope, there's all these single-celled microorganisms that are just kicking off life. So we call this the spring bloom. And if you didn't know already, phyto is just a Greek word for plant, and plankton is the Greek word for drifter. There's other types of plankton. If you're an animal plankton, then you're a zooplankton, like zoo for animal. But I study the phytoplankton, the plant plankton. And I'm going to show you this one graph here, but I'll walk you through it. So on the top, I'm showing abundance, the number of different types of phytoplankton that could exist in the water. Along the, can you guys see that okay from where you are? Along the bottom axis, I've just put chronologically different samples that were collected throughout the season. So you have November all the way through March. And then it's looking at the relative, in each sample, if you have 100% of things you counted, what's the relative abundance of different types of phytoplankton that have been found. And so just looking across, what color do you see is the most obvious? The blue. So the blue is a type of phytoplankton we call the small flagellates. I have photos of these later for you to see. And then occasionally you get a bar that has a lot of, like for instance, there's red or a lot of yellow. So those are, the red is a dinoflagellate bloom and the yellow are another type of phytoplankton called cryptophytes. And it's important to understand how many different cells are in the samples, because when you then take equations to estimate how much carbon, organic carbon biomass is available to the ecosystem from the different types of phytoplankton, what do you see now? This is showing the amount of carbon biomass on the bottom, same time frame over the season. What color jumps out to you? Is contributing the most carbon? So the gold, yeah, the gold color are the diatoms. So even if in a sample you're not counting a lot of diatoms, they're contributing a major amount of carbon. So we know that in the Southern Ocean and Antarctica, diatoms have been shown to be the primary producers or one of the dominant types of phytoplankton that we see in this ecosystem, and especially on the coast in the Western Antarctic Peninsula. And that's important because in Antarctica, it's a very short polar food web. So you have sunlight feeding phytoplankton. Phytoplankton, you learned about krill, coral, the ones Brandy finds in the flow-through system. And then krill are feeding all of the other animals. So the whales that come down to migrate, to feed, bring their calves. The seals, the penguins, of course, feast on krill. It's one of the managed Antarctic resources by the Antarctic Treaty members. And so krill is really important for the Antarctic ecosystem. But what I find interesting about krill is that researchers have done experiments to show they're selectively feeding and looking for diatoms in particular that form chains or are very, they're centric diatoms, like a circular shape, and they're full of lipids or omega-3s. And lipids are what they need to feed their young. So the young krill will go up under that sea ice and feast on the lipid-rich phytoplankton. And that is what, there's actually a cartoonist, the woman who draws this works at NOAA in San Diego at the Antarctic Ecosystem Research Division. So if you like comics, nerdy comics, you could follow her work. And I always try to check in with like what is happening in Antarctica and how might the ecosystem be changing. And if you've heard of the IPCC reports, the International Panel on Climate Change, puts together these reports that give a current state of our understanding of the world. But there's a special chapter, chapter 3, is on the polar regions. And it's a pretty long document, but just to give a quick summary, what we do know about the Antarctic Peninsula is that it has some of the fastest rates of warming recorded since the 1950s. The temperature has raised up to 5 Celsius, which is a lot for the Antarctic Peninsula. And it's not just the air that's increased, so the air might melt ice from the surface, but it's also the ocean. So that ocean coming up and hitting the tidewater glaciers is going to influence what's happening to the ice from underneath. And different studies have been done that have shown over 87% of the glaciers on the peninsula are in retreat. So that means they're not accumulating enough mass as they flow when they calve off to replace that. So the face of the glacier is moving away from the waterline, exposing more bedrock. And that is having an impact on the wildlife patterns. So where can penguins find rocks to make their nests? Where can seals haul out if they can't find ice? Where are the whales going to go find the krill? Where are the krill going to find the phytoplankton? How is the glacier melting bringing fresh water to the coast in the nearshore environment? All the conservation decisions made in Antarctica are made using the best available science. So having information about how this region is changing is really important. And in a place like Antarctica where it's very remote and difficult to work in, it's a harsh environment. You can't just put sensors in the water. Icebergs will rip them out. You don't find them when you go back to look for them. And it's very cloudy, so a lot of times using satellite data, you're missing information because there's cloud cover. So collaboration down in Antarctica is incredibly powerful to get more eyes on the ground, so to speak, or more observations that we can see. So I personally am trying to understand how can I use satellite data. So what are we able to monitor from satellites orbiting? In that, Debbie showed a cool animation of the polar orbiting satellites going pole to pole. It covers the Earth every 2 days. We get a full picture. And how do melting glaciers in the actual water affect the species of phytoplankton? And here's a scale showing you that difference between a large centric diatom on your right and compared to the smaller flagellates that might be available. So in large numbers, maybe a krill could eat those smaller ones, but in general, they want the big food. And the expedition vessels are down in Antarctica for 5 months every year. So starting now late October until March, sometimes April, there's a constant flow of opportunity to be in Antarctica, whereas as a researcher, you might get 1 month or 2 months on one of the research vessels for that 1 year you've planned your study. Because of that difference, this was the inspiration for creating a program called Fjord Phyto, which is a citizen science collaboration to engage the traveling community in science. So this project, you leave the main ship and you get in a small, one of the inflatable Zodiacs if you had a hangar tour, and you go out and you sample phytoplankton. I have a whole suite of things that are hands-on and engaging for guests to do. And this is a partnership with the industry that can help gather, every year it's about 100 samples or 100 sampling events that we get back. So it's currently funded by NASA. We've convinced them we're ground truthing what the satellites see, which is an important thing because satellites still need to be calibrated and ground truthed with in-water measurements. So it's not just me that's doing this work. Actually, we have a team. So there's Martina, advisors on the team, and then over the years, we've worked with different filmmakers, artists, undergraduate students, social scientists to try to expand the impact of this project, not only on the science side, but also in the outreach side. So I wanted to show the team there. And I wrote about how I designed this program because I thought it was important to put in an oceanography magazine, which is a pretty wide-read magazine by other scientists, to try to inspire them to think about other ways to do science outside of the lab or outside of big research vessel style investigations. So what can we do to learn more about these places together in a collaborative opportunity, but also that allows for a lot of impact. So, getting people engaged. I said I dropped these instruments over for people, so we assemble a kit every year. That kit is on board now, and the kit has various oceanographic instruments that are handheld and user-friendly, and I'll kind of show you the video as I walk through. So there's a device that can be dropped in the water column down to 100 meters, and it measures the saltiness, the temperature, and how much chlorophyll, or that pigment that phytoplankton have, or all plants, in fact, where the signals are coming down from surface down to 100 meters. And then we drop a Secchi disc, which is just a circular disc that can give you a quick estimate of the amount of light that's getting through the water, or how turbid the water is, how much growth there might be from phytoplankton. We also drop down a phytoplankton net. So for 10 minutes, there's a net that gets towed behind the boat to concentrate the amount of phytoplankton. That then gets filtered back in the lab or on the boat. It can happen in either location. And then that filter gets preserved, and that's how I then take it into the lab and extract the DNA or the genetic information from the sample. And then we also take a surface sample. That is how Martina can count how many, quantify how many cells are in that volume. And then we also want to know if we do see a freshwater layer on the surface from the CTD, which stands for conductivity, temperature, and depth. Does the CTD show us a freshwater layer? Another sample can tell us by oxygen isotope analysis if that fresh water was sourced from glaciers as opposed to sea ice. So to give you a quick visual of what a cast might look like, this is showing salinity, or the saltiness. So on the bottom, it's just a scale showing 33 up to 34.5 PSU, which is just units of measurement for salinity. And the two different locations on the left are showing a freshwater layer at the surface. So when you're at the surface, the reading was fresher, but then when you go down to 5 meters, you see that it changes drastically, and then the whole rest of the water column is the same salinity. Whereas you compare to another location on the right, and you see that the surface down to almost 55, 60 meters is well mixed, but it's a well mixed fresher layer. Then you see it change and it becomes saltier. In contrast, you have a similar profile showing temperature. So these same two locations, the temperature was much cooler, is negative 0.5 at the surface, and then it has a little blob of warmer water. Warm is 1 Celsius, and then it goes back to being slightly cold at 0.5. Whereas on the right, I'm like, what is going on with the different layers of water? There's a layer that is negative 0.5, but then there's this blob of warmer water, or negative 0.3, and then it gets really cold down to negative 0.7, around 60 meters. So I don't know what's happening or why the circulation is acting like that, but using the salinity and the temperature, you can also say something about the density of the water. So those are different water masses that are sitting on top of each other, which is why they have unique signatures. And when you analyze the physical oceanography, you can then tell something about the environment. That chlorophyll sensor I told you about, so same two locations. The chlorophyll, the scales are different here. So on the left, it's reading at the surface 0.2 micrograms per liter is the units for chlorophyll. And then you have a big spike of chlorophyll. So in that layer where there's a big spike, there might be more phytoplankton growing. Maybe that's where we saw the Secchi disc disappear. Maybe that's where it'll get, the phytoplankton prefer living in different layers. You're not too close to the sunlight at the surface, but not too deep that they can't get access to nutrients. Whereas on the right, the chlorophyll from the surface down to 55 was in that well-mixed layer, but it's also, the scale now is between 10 and 20 micrograms per liter. So that's a lot of chlorophyll in that particular location. And I have to figure out why. So to just show you a visual, the difference between the net sample that was collected, when we concentrated for 10 minutes, the water still looked pretty clear in that jar. Whereas in the sample with high chlorophyll, it was a nice dark brown, greenish collection. I was curious what the satellites showed. So we have one of our team members pulls the satellite imagery and makes composites. So because it's so cloudy, you don't always get, like every day, you can't see what the surface of the ocean is doing. So you see, for instance, in the left, there's a big band of gray. That's because probably there was clouds there, and so it wasn't enough data over a week to put together the composite of chlorophyll. But I found another one that was actually 2 weeks earlier that did have a clear composite. So this is in the red circle. I'm showing that if it's cooler colors like purples, that's low chlorophyll to no chlorophyll or no phytoplankton. But if it's getting into the greens, yellows, and reds and oranges, that's a lot of phytoplankton. And the scale on that was also around 10 to 20 in the oranges and reds. So I thought, I'll do a deeper analysis, but just when you're on the ship and you could see that, it's like, oh, we just collected this, it's really brown. And the satellites also show that there's a lot of chlorophyll. So, that's one of those ways to ground truth that data. And that's also important because when you look at it, as I said, you can have a lot of chlorophyll, a lot of phytoplankton, but if you're a krill and you're selectively feeding on certain types of phytoplankton, you look at under the microscope on board here and you can see there's completely different communities of phytoplankton living in these different samples collected from different locations in Antarctica. So it's important to understand what the different species are. As you can see, chlorophyll can tell you bulk amount of food, but to really understand the carbon that's available, you also want to look at the species that are present. One of the cool things we saw in March, March is getting into the later summer, so the sunlight's starting to disappear, you're starting to get darkness. Winter is coming, so we saw some of the phytoplankton are actually now transforming into cysts. So that's kind of like some of them go into hibernation phase. They'll either get frozen up into the sea ice or they'll turn into cysts and sink to the seafloor and just wait it out till spring comes, and then you get the water upwells and the nutrients come and then they can thrive again. So this is a certain type of Diatomacatoceros that is forming horns, and it's just going into that protective casing. And a study was found some spores in a sediment core. And the researchers were actually able to date the different layers of sediment and look at the different types of spores, and they come in really weird shapes. It's like very alien looking. And the different spores, they were able to successfully germinate a spore that was dated to be 7,200 years old. So think about that, because it's like if things change in the environment, phytoplankton have a way to just sink down, and then it could be thousands of years before they could come back to life, or who knows what their plans are for their future. But I thought that was pretty impressive to see that they truly are surviving. So this is another one of the samples showing the different phytoplankton, and then those samples this is one of the filtered samples that gets concentrated and then sent back to me in the lab in San Diego. And in San Diego, then I'll take all these samples, I'll take out the filters. I don't actually get to really see what they look like under the microscope. So Martina's here with me now in the US. She's been with me this summer. The Antarctic Tour Organization, or the International Association of Antarctica, tour operators provides fellowships to scientists, and so Martina got a fellowship pre-pandemic that she had to wait to actually come to the US this summer so we could look at some of these samples. I get to see bands. A white band means that there was DNA that was extracted, and the gene that I was interested in, which allows me to identify species, showed up. So that is you want to see bands. And then Martina and I are working on looking at individual species that were of interest in different samples. She gets another portion of the sample, that collection of surface seawater, and she uses microscopes. So this is showing she takes a sample, she needs to let it settle for 24 hours, and then she's ready to look under the microscope. It can take her anywhere from 3 to 5 hours to count one sample, and she's looking at the yellow image like that. So it's not always like a beautiful, glamorous job, but I feel envious that she gets to see what is living in there, whereas I just get brown or green goo. She also has a scanning electron microscope, so she gets to put them under this really high-powered microscope to get these images, which is then how a taxonomist can just look at the shape or morphology of different species and try to identify it based on like very nuanced things, like if the stripe is straight down the middle or how many dots there are, or if the dinoflagellate in the middle had the waistband going this way with a little notch, or the notch was to the left slightly. And then it's some more images of those cysts. So we don't know what the cysts, it's hard to identify which species has turned into the cyst. There hasn't been a lot of work on matching those up, but that's how we get some more detailed information. Just some quick results that have come from this so far. We've published in Polar Biology. We're trying to do open access scientific journals because as a citizen science project and wanting this to be available to the public or anyone to be able to read, we are looking for journals that allow for that. And in one of the samples, we documented a dinoflagellate bloom that had more than 6 million cells per liter of water, which is a lot because most researchers come down thinking the diatoms are the most important. But when you get these chances, because the expedition vessel's sampling through those 5 months, we can actually see when these blooms might happen that other research projects had not seen because they're doing snapshots in time, whereas we're getting more of this succession over the whole season. There's a species that we found that hasn't ever been observed or documented. So that's what Martina and I are working on. Pulling out a single cell, extracting the DNA, and just trying to see what is that one individual, because she's looked under the microscope and she can't identify it. So that could be cool contribution to science's new species of phytoplankton. Maybe we'll name it after, I don't know how you name phytoplankton, but maybe it'd be like the dinoflagellate Viking or something. And I have a colleague, Christian, who helps me in the lab process these and do the genetic side of things. He's actually going to get to come on board in January to work on figuring out if, there's genetics capabilities on board. Can we make genetic sequencing happen in the moment? When we're in Antarctica, because that could offer an opportunity. If you see something unique or out of the norm, you could collect more samples if you had an ability to identify that species right then and there, or see that it was missing from the database, and so you want to collect more material. So on the top, I'm showing the size of plankton. And remember I said there's phytoplankton and zooplankton. So this is showing all the way down to the size of viruses, which there are tons of viruses in the ocean. And they eat phytoplankton, they attack phytoplankton and bacteria, and then it goes all the way up to jellyfish. So in the surface sample we collect, we don't get jellyfish, we don't get krill, they're far too big and fast for that. But we are able to see the smaller range of sizes of life that exist, mostly single-celled organisms. And then when we do the phytoplankton toe, that's a mesh size that's 20 microns. So if a human hair is the width of around 100 microns, then this net is just targeting the size of what we call microplankton, so just based on the sizes of them. And those protists are then what we're extracting the DNA from and comparing to the protist databases that exist online. I gave a talk for the Scientific Committee on Antarctic Research earlier this year, and I wanted to share what my side of the data looks like with you guys. So I'm just going to show you what I learned about the diatoms over the first 3 years. So just comparing the 3 different seasons, partial season in 2020 because of the start of the pandemic, everything kind of got scattered around the world. So I've had to wait a couple years to get everything back. So I'm currently in the lab finishing processing the rest of 2020 and then last year's samples. But if you look at genera of diatoms versus species, I can start to compare to the databases how many different genera we're finding. In total, we've actually found up to 180 different types of phytoplankton in the samples that are just collected from the citizen science program. And then I just made a word cloud to show all the Latin words because it's science communication. How else do you show all the diversity that you're finding? There's many ways with statistics, but another way I can look at this is more of those colored bar plots. So this is showing if I have that filter and I extract the DNA, and then of the DNA, I can attach barcode or like a gene. I'm just looking at one gene that's conserved through the tree of life. So when you compare it to the databases, it's like if any change off that sequence is different, then it can assign it to that species or try to get down to a species level. And then it tells me, okay, that's the brown color. So brown is Corythron. And I could go down to species level if it had a match to the database that it was confident, above 98% confident that was what it thought the species was. So I just see a bunch of colored bars, and it's showing, again, a relative abundance of, this is amplicon sequence variants, so that gene that I amplified. And this is, again, over the season from November through March. And so I go and I ask Martina, what do these look like? So she says, oh, the brown, there's Corythron. So it's this cool diatom that has a bunch of spines and spikes all over it. Then as the season's progressing, it turns into this purple color, which is the chain-forming diatoms. So that's the ones that krill tend to have a preference for, and that is Porosira. As we continue, there's another chain-forming diatom that pops out in the yellow. That is a Thalassiosira, another omega-3 lipid-rich diatom that krill would like to feed on. And now around this time of year, we're getting into January. It's the peak bloom. This is also, after January, the whales have started to arrive. They're looking for that krill because the krill have had the beginning of the season to be eating. Then we go into this bright green. That's a Eucampia, another type of diatom. Then we go back into the browns and into this purpley color. So there's the disc-shaped ones called actinocyclis. So it's like, what does it all mean? Why are they all there? How does this compare to what Martina's finding in the microscope samples? How does this compare to the environmental data that we got from the satellites, but also the CTD that we dropped in the water? Was there fresh water at the beginning and in the different locations, or fresh water that came later in the season, and it might have shifted the types of phytoplankton available? So this is 2017 and 18. And now I'm just going to show you the following year. Just by color alone, you can see it's totally different, right? Like, why is that? What is the difference? I don't know yet, but I will soon. So in this case, it's completely dominated almost every sample by that purple color. The season starts out with this Eucampia green, which those guys didn't show up in the previous year until kind of late December, early January. But now they're starting off the 2018-2019 season in the middle of November. And then you have the pink is another type of centric diatom that's smaller, called Chionodiscus. So what happened from the prior year that is why this succession is starting off this way? Again, your Porosira is that purple bar that is just dominating almost every sample. And then you have this orange spike in the middle here. There was a giant bloom of a type of diatom called Catossaurus. That's the one that was going into a spore. And these to me look like centipedes, but when they grow, all their little long spines kind of interconnect, and that helps them float at the surface, so they make these big mats. But it's like what was happening in that location on that day. That's something I can look at. And then the end of the season, this other lavender color, purple towards the bottom, Proboscia. Like, why did they show up? So it's not just over time that it can look, it's also now piecing out different locations. So if we have the whole peninsula, and I'm just comparing 2 locations over 2 of those different years, I can just quickly see that there are differences. And then we can do testing and statistics to see what the diversity is and actually what the changes might be correlated to with the environment. So the bigger picture is that this program has been running for 5 years now. We didn't get to go down in the pandemic year, the first one. So there's a data gap there. And over time, 33 different sites have been sampled. So these are most of the sites that the ship's going to. And if you imagine that 10 people could get in a boat together, that's over 4,000 people have participated in helping collect samples for us to understand how this ecosystem's operating. And if each boat is taking a collection of samples, that's we have over 400 samples. So over the years, we've presented at different conferences nationally in the U.S. and Argentina, and now with the online world, we've presented at places we might not actually ever go travel to. And we're also, because Martina is from Argentina and a lot of the gateway ports to Antarctica are from South America, we're doing bilingual conferences, so in Spanish and English, which I think is cool to like try to reach a broader audience even. So just to summarize the impact of the Fjord Phyto Citizen Science Project, looking at phytoplankton in the fjords of Antarctica, with the ability to have the collaboration with the expedition vessels, we can get more of a succession or how things are actually changing through the season with that frequency of sampling or monitoring. Instead of traditionally with research, it's just snapshots in time and trying to compare like what did we see in December versus April. Instead, you can see the whole change happening. And additionally, I'm looking at, are there genetic advantages between different species? Like, why did we see some of those diatoms in different fjords? If it was, there's a hypothesis in the literature that's showing in fresher water, you get a shift towards the smaller phytoplankton, but that's not conclusive. So could this project provide that evidence? If that's the case, then that could be a problem for krill. If fresh water is influxing into the coast, but then you're shifting the amount of food, carbon available because of the species of phytoplankton now are changing. I want to get at why is that, not just the correlation. Is it correlated, but like, is there a genetic reason that's driving those differences? And then another cool thing is that the databases, as I mentioned, may not be what they seem. So Martina actually found, she was working in one of the fjords prior to Fjord Phyto starting, and the database said that the sample was 98.7% that it was this image that's shown on your right, Triceratium dubium. And she's like, there's no way that's what it is, because that is a species that was collected out of Hawaii. So there are species that are cosmopolitan, they live globally around in the oceans, but that is definitely a Hawaiian species. So she showed an image under the microscope of what she's seeing, which is Odontella westflagii, which wasn't in the database. So by combining our approaches to analyzing these samples, we're hoping to inform the databases of the different polar species and kind of build up that data gap. So that is Fjord Phyto, and I want to admit something to you guys. I actually always used to think the ocean was really boring. I was like, whatever, it's just a big body of water, nothing's really going on. And over the years, I have learned so much about phytoplankton that now that's like all I can see when I look at the ocean and how cool it is. And just to kind of think about phytoplankton as floating solar panels scattering on the whole surface. I get asked often by young kids when I give talks to schools, they're like, what do phytoplankton eat? And so, you could imagine it's like a plant, so it needs light, carbon dioxide, basic nutrients like you would put fertilizers on your plants at home. And Brandy talked about this morning the need for iron in those zones that don't have a lot of iron. Whoops. And then your job is to just float around and not die. So what was the first plankton or photosynthesizer? I already gave it away. I didn't set this up right. It's a cyanobacteria. So I imagine you guys have heard of blue-green algae or cyanobacteria. So that was an organism that was shown in the fossil record to be one of the first organisms to figure out how to photosynthesize, how to use sunlight for energy. And when we look at the water, we're looking at that amount of sun or the visible spectrum that's coming to the surface of the water, and then it's showing a depth from 0 down to 100 meters. So the light level tends to drop off around 150 or 200 meters. So that upper zone is where you get light, but you get different wavelengths of light that are making it deeper into the water. So a phytoplankton's preferred zone of living is actually between 30 and 60. That's where we tend to see that big spike of chlorophyll pretty often. That's like the sweet spot for them. But cyanobacteria were just a single cell that, if they figured out how they could use the light that's coming in and then use carbon dioxide, which was abundant in the ocean, and then they use that for sugars and energy, and then they spit out oxygen. So it's actually a waste product for them. It's toxic to them in large numbers. So now you have oxygen that's being put in the ocean and kind of building up over time. And when people look back at the geological record to see when did this happen, it's around 2.7 billion years ago. So the origin of cyanobacteria is being shown in the fossil record. And then it progresses to being able to diversify into other types of single cells as they're evolving and trying to survive. Eventually you get enough oxygen built up that you have something called the Great Oxidation Event, where there's enough oxygen in the atmosphere that you could then continue diversifying. And then you get the first eukaryotes showing up, and then eventually first soft-bodied macrofossils, then of course the charismatic dinosaurs. This is shown in a different way here, which on the top panel is a scaled out view, so of time over billions of years. You have the origin of life happening. On the y-axis, it's showing the atmospheric oxygen levels building up. And so you can see it was like no oxygen, around 3.7-ish, you get these cyanobacteria. And then where I've circled in red, that's where the oxygen just went up. And you had enough oxygen, which is scaled in on the bottom panel. So again, atmospheric oxygen, and you can see it's kind of oscillating around 20%. And so you get the plants. The Cambrian explosion was that explosion of life in the ocean. There was enough oxygen going on. And then plants actually came out onto land 420 million years ago, and then you get the rest of life. So these weird little single-celled organisms that we don't give much thought to decided it was better to live together and to photosynthesize. And so they have the ability to just divide and split and grow, so they started populating the ocean. And we call that endosymbiosis, when you can live together for a different cause of life. And this is where I really get into the phytoplankton and think it's like sci-fi going on. So if you had that first cyanobacteria event where one cell engulfed blue cyanobacteria, that gave rise to green algae and red algae, and then another type of plankton or protists that you don't have to worry about. But green algae are what gave rise to all the land plants. So every land plant is originated from a green algae in the ocean. And then red algae, the two of them continued to live in the ocean and get engulfed by other cells, which then turned into an endosymbiosis event. And it kind of radiated out into all these different species or types of phytoplankton that we have in the ocean. And some of them are insane. They have genomes that are 100 times larger than the human genome, and I'm just like, why do you need all that information? But it was really hard to piece together how these are related in the tree of life, because they're like Russian dolls all stacked in with each other. So it was kind of like the research team that likes to investigate this puzzle. I appreciate the final figure, but I think it was like a scavenger hunt. And it's a whole category of life I feel like doesn't get much attention, but they're called the protists. So they're actually not plants. They're not animals. They're not fungi. They're also not bacteria or archaea. They're this whole other existence in the tree of life. So protists, or phytoplankton if they're floating in the ocean. So I'd shown you this, that the amount of light getting into the ocean, if you're a plant or a phytoplankton, you have chlorophyll A, but other types of phytoplankton are trying to find their niche and trying to diversify themselves. So they're looking to use the other wavelengths of light that are available. So they start to create accessory pigments. So they'll have chlorophyll B, chlorophyll C, carotenoids, phycocyanin, phycoerythrin. They're kind of building up a suite of pigments that can harvest different wavelengths of light because they're these energy factories. They want to be able to use that efficiently. Some of the phytoplankton have even gone into kleptoplasty, so they have this ability to cast a mucus net around another group of phytoplankton and digest them with these enzymes and slurp them up, but keep the chlorophyll or the chloroplasts and then use it for themselves and float around, so they don't actually generate chlorophyll themselves. They're stealing it from other phytoplankton. That's one strategy of life. They're like, didn't need to live symbiotically, they just go around thieving. One of the things that we can do traditionally with phytoplankton is look at those different pigments. So this is just to show you, if you notice, the purple on the left is the cyanobacteria. So they're the different suite of pigments that would tell you it's a cyanobacteria if all you were looking at were pigments in your sample. The greens are in the middle, so the suite of green highlighted ones tell you that it's prasinophytes versus chlorophytes. And then if you're derived from red algae, you're in the red highlighted category, and the dinoflagellates, chrysophytes, diatoms, cryptophytes, primnesiophytes, they all evolve from the red algae, so there are different suites of pigments that indicate what you can see. This will come in handy later, so remember just that you can categorize phytoplankton based on the suite of pigments in their cells. So we went over the basics of what phytoplankton eat. Also, they need to reproduce to survive, so they can just do that by splitting themselves asexually. But you could see a problem with that. If you're cloning yourself, then your genetic diversity is going to be compromised if anything came through and threatened your population. So they sexually divide. They have sex. That's weird. It's a single cell in the ocean. So they'll release gametes, and the dinoflagellates and the diatoms life cycle has been shown in detail. And when they're growing, whether they choose to do it asexually or sexually, we call that a bloom, so they're rapidly dividing. And so the color changes in the water, and those blooms are individual phytoplankton species, but they can be seen from space. So this is where the satellites are looking at ocean color, how the ocean's changing color. And so this is like, if you guys have seen red tides, the red tide is just a phytoplankton that has reddish pigments. So when it's growing in large numbers, it's changing the color of the ocean to look red. So satellites that's what they're trying to detect, are the chlorophyll, but also could there be other pigments that are detected? And can we get a global scale monitoring of this? Right now I want to do a little thing with you guys. I want you to all inhale deeply with me. So that breath, like I said, phytoplankton have a waste product that we love, oxygen. You may have thought that most of the oxygen in the world comes from plants and trees on land, but if we take another breath and exhale, that second breath came from phytoplankton. So it's estimated that 50 to 80% of Earth's oxygen is generated from phytoplankton, depending on if you're measuring from a coastal zone, upwelling zone. And I love this image. It's a composite of the chlorophyll on Earth from satellite imagery, showing the seasons changing. It's summer, then it's winter in the northern hemisphere, while it's summer in Antarctica, and it's the Earth breathing. And so it's the chlorophyll on land and in the ocean. It's mesmerizing. It's all because of phytoplankton and plants. So one of the other things that phytoplankton have to do is avoid death. Nobody wants to die, not even the phytoplankton. So they have to think about how they're going to get nutrients or not eat up all the nutrients around themselves. They actually form relationships with bacteria, so the bacteria will live with them and provide certain nutrients like B vitamins that they can't make themselves. They have to avoid being attacked by those viruses. Viruses are everywhere, and it's one of the things we think, this was the study in the Ross Sea, is maybe as the algae that's growing sinks, maybe the viral attack is helping break up that carbon so that other bacteria can eat the carbon as it's sinking to the seafloor. So viruses might actually have an important role in regulating these cycles. They want to avoid being eaten or grazed, so they form like cactus, form spines to keep different plants have thorns. Phytoplankton have tried to have strategies to avoid being grazed upon, or they produce toxins, which is why some of those red tides produce toxins that can be very harmful algal blooms. It's just them trying to survive. But when they grow in large numbers, it's not good for the animal life. And then, of course, they want to avoid sinking, or they want to stay where the light is. They don't want to get too heavy and sink, so a lot of them will raft up. That's what the chains are. Two are making a lot of lipid, helps you float in the surface where the sunlight is. So that's a lot about phytoplankton. And we get asked, how is it going to change as the Earth as a whole is changing things in the ocean? This is showing the red tide. Some of those red tides bioluminesce. So these tiny little microbes are responsible for large-scale phenomena. And one of the cool things that's happening is a new satellite mission called PACE. Has anybody heard of PACE? So it's, Debbie has. So it's sensors that are going to be launched in looking at plankton, aerosols, clouds, and ocean ecosystems. And instead of traditionally we've had different spectra sensors. So for instance, the images I showed you with the chlorophyll was from VIIRS on one of the satellite ships that had 22 spectral bands. The PACE mission is planning to have full spectrum. So that's what that bar is showing, from ultraviolet all the way down to shortwave infrared. So instead of seeing segments of different ocean properties, we're going to be able to see a lot more. And that comes in handy because that's where the different pigments that you could detect, maybe these new sensors, you don't just get chlorophyll. Now maybe you'll be able to distinguish those groups better and where the different phytoplankton groups are growing in the ocean. So other ways we're trying to image phytoplankton just in general is, are there ways to, instead of like Martina counting for 5 hours one sample, could you get instruments that will do it automatically and be able to, like facial recognition, like recognize which species are which? In that sense, for coastal zones, which harmful algal blooms are a problem, you could have an automated system to characterize which ones are good in the system and which ones are the toxin-producing phytoplankton. So that's just some cool ways that we're trying to understand phytoplankton better. A couple more ways that phytoplankton could help us in a changing system. Brandy talked about iron being limited in some parts of the world, so there's been a lot of efforts to fertilize the oceans by dumping iron in them. So if you dump iron in, you trigger the phytoplankton to grow. And maybe you could escalate the amount of phytoplankton sinking to the seafloor, which is where we're getting all our fossil fuels anyway, is ancient phytoplankton from the past. Maybe we could speed up that sequestration process. But there's problems with that because, as we know, with harmful algal blooms in coastal areas, sometimes then you get this negative effect where all of the oxygen has been depleted, and then you get animal die-offs, bird die-offs, fish die-offs. So it's a little tricky to engineer how phytoplankton are going to respond, especially when we don't know very much about them. And then finally, you could also use algae for biofuel, so genetically engineering algae for which genes make them grow really rapidly, and finding the algae that do well in outdoor raceway ponds, and then being able to harvest the energy directly from them. So I hope that over this last hour, I've shown you how single cells, or what I like to call the invisible forest, can have a huge impact on global cycles. And so even though they might seem microscopic and kind of boring, there's actually a lot going on in the phytoplankton world, and we are coming into port right now too. So I just want to leave you guys with that mesmerizing image, and thank you for your time.